Thanks, Nick, and I'd also like to thank Sebastian Lunke, the principal investigator of this project, and his co-lead, Zanitza Stark, for supporting me to present our project today, which is BabyScreen Plus. So I just want to start by setting the scene for what is happening in Australia more generally. So you can see here there's a number, 5 projects that are looking at genomics in the newborn screening space. These are federally funded projects, and they come together in another consortium, GenScan, to have a look in a more local context, how we can share those findings and maximise the impact from each individual project. And there's actually quite a robust Australian cohort here, so you'll be able to learn a little bit more about not only our project but the others listed here. And I'm not sure if I'm allowed to use this platform for this, but if the Australians want to gather under the sign at the start of lunch, maybe we can grab a photo for this particular consortium. So BabyScreen Plus is a pilot study, so it's really looking at the feasibility of our process and the acceptability to parents. We are doing newborn screening using clinically accredited whole genome sequencing, and we're going to sequence 1,000 newborn babies in the state of Victoria, which you can see in red here, in the southeast of Australia, and we are actually actively recruiting at the moment. So our study process has really been designed to be scalable and to align with our standard newborn screening process. So we've made the decision to offer the testing during pregnancy, and it's going to be performed alongside the standard newborn screening. So we're very lucky in that we're using the same sample as standard newborn screening, the heel prick sample, and we've got this unique situation where the laboratory, the Victorian Clinical Genetics Service, is housed where our standard newborn screening test is done, just a few doors down from where our genomic testing's done. So we're very lucky in that respect. The test, as I mentioned, is actually clinically accredited whole genome sequencing, so the results can be used to feed directly into clinical care. And the results turnaround time is 4 weeks after the sample is collected, so around 2 weeks after that DNA is received in the lab. Our result return is either low chance, which is returned online, and I'm going to talk about that in the coming slides, or high chance, which is returned by a clinical team of genetic counsellors, clinical geneticists and disease experts. So I just want to go a little bit into the consent. Again, we've really focused on making this scalable. So genomic newborn screening's being offered at about 32 weeks gestation, and it's via the Genetics Advisor platform. So this is a heavily evaluated platform that's been used in other research studies and other settings, and it's been adapted for this newborn sequencing setting. So it's a completely online process of enrolment, education and consent. So women can just scan a QR code that they're provided for from the obstetrician to access this platform. They're able to provide 2 levels of consent, so research consent to participate in gathering data for BabyScreen Plus, and a separate component of clinical consent to have that accredited whole genome sequencing performed for their baby.
What is newborn screening, and what can I learn?
A video here.
Every new baby has a test to screen for genetic conditions at birth. This is called newborn blood spot screening. This is performed on a few drops of blood collected soon after birth by pricking the baby's heel. Newborn screening looks for rare but serious health conditions in babies. These conditions can be life-threatening or affect development. New technologies can be used in newborn screening to look for many more health conditions. One of these is genomic testing. This is a powerful tool we use to look for DNA changes that can impact health.
So I'm delighted to see that although we can get very distracted at conferences listening to people talk, and even in one-on-one conversations, I noticed that's a very engaging sort of process. Everyone was looking up and interested in what was going on. So we think that this kind of platform is scalable, but also provides that level of engagement required for informed consent in this setting. Families do have access to a hotline where they can talk to a genetic counsellor if they need more nuanced discussion about whether they want to participate. But for the time being, that hotline has not been utilised a lot, and this seems to be a very effective way of consenting parents. So the next big topic I want to move onto is what conditions we're screening. And so very similar to the projects we've been hearing about, we're looking at severe conditions, early onset, which we've classified as less than 5 years, treatable, but also really importantly to us, because we are doing a clinically accredited test, the conditions needed to have, and genes needed to have analytical validity and gene disease validity. We're not reporting carrier status, adult onset forms of the conditions, or variants of uncertain significance. And our multidisciplinary team to come up with this list was comprised of clinical geneticists, genetic counsellors, lab geneticists, and disease experts. Perhaps slightly differently to some of the other projects, our gene list is available on an open source platform, so you can put into your phone Panel App Australia and you can have a look at our gene list and also all the rationale that went into the genes we've included and excluded. So you can see there how many people have reviewed it, the comments they've made, the literature they've drawn on. So it's a very transparent process and one we hope can be built upon. And really interestingly, there's been some discussion about this. We've also had a look at how our list has compared to all the lists of the projects that are being presented here. And you can see from that amazing and complicated Venn diagram that there is very disparate outcomes, even though we are all working with similar criteria. And some of this we think comes down to that very different starting point. So as I mentioned, we've really focused on things that are relevant for a clinical grade test, but others, for example, the BabyScreen project have looked at gene disease validity as a starting point, compared to, say, Beginnings, which is maybe focused more on treatability. So for our study, we've come up with 604 genes, and honing in on what that Venn diagram is trying to demonstrate, there are only 55 genes in common, so less than 5% when we looked at all these different studies. This slide is just demonstrating the really comprehensive evaluation we plan to do, and I think this demonstrates how we're trying to squeeze as much as we can out of a relatively small cohort. You can see our evaluations in 3 phases: pre-implementation, implementation and post-implementation. And although recruitment's only just begun, we have already started this evaluation process. And you can look out for a couple of publications that are in the works around key informant interviews, for example. So in summary, we are now recruiting. Of the 1,000, we've recruited about 100 mothers and we've had about 30 results issued. We've had a very high uptake with that offer in late pregnancy, and all reports have been issued within our timeframe that we were aiming for. We've only required 1 recollect of samples, so it has been a smooth process so far, touch wood. And we have been recruiting so far through 1 private practice, just as a slow start, and we've just opened up to 1 regional hospital and plan to engage another large public hospital to get those numbers. So I guess the big picture for us is that we really wanted to focus on a scalable and user-friendly system. And we're really focusing again on a multifaceted evaluation so we can really maximise our outcomes from this group. And I've put a picture of a building here that you will see if you make the very long trip to Melbourne. This is in Federation Square, so a very public space. And while it's a cohesive building, not everyone will like it, but it is a cohesive and functional building. And you can see it's got that patchwork effect. And I relate that to our project because we've really focused on building on previously laid foundations. So things we've learnt from this group and from other research and from previous work we've done in different settings like acute care genomics to try and have a really strong and cohesive project that we get maximum benefit for our families out of. A really big team to thank and a wonderful team to work with. Thanks very much. Even with the use of my video, I do have time for a question if anyone wants to ask one about the project.
Microphone. So wait for the microphone because otherwise streaming people can't hear.
You know, this is kind of a critical question in my mind. You said you're going to focus on, early onset, not late onset. So what do you mean by that exactly? Are you not going to call out a baby that has a severe pathogenic and a late pathogenic variant and therefore is predicted to have a late onset disease for autosomal recessive diseases? Are you only going to call out babies that have severe/severe? How are you going to avoid the adult onset?
Yeah, so I think there's 2 layers in which we have tried to avoid giving parents information that is not relevant or able to be acted on at that point in time. So I think these parents are at a vulnerable time. They've just had a new baby, and the layers that we've put in to, I guess, protect them from harm are both selecting conditions and gene disease associations that we know mostly have onset in childhood less than 5 years of age, and we will also do that at a variant level. So if we saw a variant where onset was only known to be in adulthood or very mild, we would not report that.
Very good. Okay, thank you for a clear answer.